In this lesson, we're going to look at one of the key ways of amplifying DNA samples. So before we can get into how we amplify the samples, we need to review what DNA itself is. So to throw back to year 11, we know that every DNA strand is made up of a whole bunch of molecules known as nucleotides. And we also know that each nucleotide is made up of three key components, the first being a phosphate group. We can see that in our diagram in pink. Secondly, we have a deoxyribose sugar, so a 5-carbon sugar, and again we can see that in green on the diagram there. And finally, represented in yellow, we have a nitrogenous base, and in this instance we have adenine. However, there are 4 nitrogenous bases, so we have adenine, thymine, guanine, and cytosine. So when we look at these nucleotides, it's really important that we recognize that they form bonds together between the two phosphate groups and the sugar. And this is what forms the sugar phosphate backbone. So I'm just highlighting this in yellow, running down the left-hand side of this DNA molecule right now. So this is what we call the sugar phosphate backbone. The second important part that links all of the nucleotides together now is what we call the hydrogen bonds. And the hydrogen bonds are the bonds that exist between complementary nucleotide pairs. So cytosine and guanine, and adenine and thymine in DNA. Now, thanks to the sugar phosphate backbone and thanks to these hydrogen bonds, the DNA ends up forming a structure known as a double helix, where 2 complementary strands run parallel to one another and are twisted around one another in a helix shape. So that's our very quick recap. Let's jump into what PCR actually is. So PCR stands for Polymerase Chain Reaction. And you've probably heard of PCR before, especially during COVID times with things like PCR tests, where the purpose of these tests was to amplify small segments of DNA, make copies of them, so that we can then use these copies of DNA in order to test them and analyze them. So PCR occurs over 3 key steps. Firstly, we have a denaturation stage, we have an annealing stage, and finally we have an extension stage. All of them have very specific temperatures and specific jobs, and at the end of this lesson we'll go through and we'll analyze each quite deeply, but for now we're just going to go through some of the ingredients and some of the key parts for each stage. So what do we need for PCR? Firstly, you need a particular DNA sample, so this could be a particular gene or a particular DNA sequence of interest. The second thing that we need is free DNA nucleotides so that we can build a complementary DNA strand. We now need a buffer solution, and the chemists among you will know that a buffer solution is used to help us maintain an ideal pH in the environment so that this DNA replication process can occur unimpeded. Fourth, we need some primers. We need both forward and reverse primers. And what these do is assist the DNA and assist the free nucleotides with a place to start, okay? And assist Taq polymerase, which we'll talk about in a second, to initiate that DNA replication process. Finally, we have a polymerase, and the one that we use in this particular instance is Taq polymerase. So Taq polymerase stands for Thermus aquaticus, and its job is that it's a heat-stable DNA polymerase, meaning it won't denature when we increase the temperature during the PCR process. So a little bit more about Taq polymerase itself. I've just mentioned that it's heat-stable, and Taq polymerase is stable to about 96 degrees. Okay? If we increase the temperature any further, it would cause the denaturation, and then the enzyme would change shape and it would no longer work. But fortunately, this is significantly higher than what we have with other DNA polymerases, especially the DNA polymerases in our human body, for instance. So the reason that we use Taq polymerase is so that we can run this PCR process in an ongoing manner, and we don't have to add new polymerase every single time in between because it just doesn't denature. So here are our 3 key stages, and we'll break them down nice and simply. So up the top we've got our ingredients, so we've got our particular DNA sequence, we've got some DNA primers, we've got some nucleotides, and what we're assuming here is that we have our buffer solution as well. So when we're looking at the first stage, denaturation, it's really important that we note that the temperature of denaturation for PCR is 95 degrees Celsius. Now you will see some ranges here, so I know in this diagram I've got on the left it says 94 to 98 degrees. However, from our perspective, looking at the WACE exam, we're really interested in 95 degrees Celsius. Now what happens during denaturation is we have the separation of the two strands of DNA. We have this as this temperature allows us to break the hydrogen bonds between complementary base pairs, and the key part for us is that this is going to form two single strands of DNA. We can see this represented in the diagram on the left, where we originally have our double stranded piece of DNA, and now we have two single stranded pieces of DNA. Now we can use these two single strands in our next process, annealing. So the first thing that we do is we cool the temperature down to somewhere between 50 to 60 degrees. There's a little bit of variation here. And this cooling allows hydrogen bonds to form, specifically between primers and the two single DNA strands. So you might remember that we have the forward and reverse primers, and they attach to the opposite ends of the DNA sequence. So if I annotate my diagram here with the 3' and 5' markers, you'll see that each primer is going to be targeting the opposite end of that DNA sequence, or the complementary portion. And with the primers attaching, this allows both ends of that DNA molecule to be copied. Finally, now that we've attached the primers, we've annealed the primers onto these individual DNA strands, we warm the temperature up ever so slightly to 72 degrees Celsius. Now this is the optimum temperature for Taq polymerase, the enzyme that we spoke about previously, to add complementary nucleotides to those single strands of DNA. Now Taq polymerase is going to start at the primers, and by the end of this process it will create a new strand of DNA. Now, the great thing with this process, as I mentioned with Taq polymerase being its optimum temperature of 72, but it doesn't denature up until 96, is we can run this process non-stop without having to add more polymerase to this reaction. And so generally speaking, we're going to run this process about 20 to 25 times. And so this is what we call a chain reaction, where we can have very large amounts of DNA produced in quite a short period of time. Now, if you're ever asked to calculate the amount of DNA produced by this, we can simply go that's a bit of a chunky pen, let's just fix that. So we can go 2 to the number of cycles. So let's say we ran this process 20 times. You could put into your calculator 2 to the power of 20, and that will give you the number of DNA produced in that process. So I've got another representation here. I'm not going to go through this one right now, but if you're interested, you can pause the video here and have a bit more of a look at this diagram. It's just another representation showing these 3 distinct processes. What I will do now though is I'm going to jump down to some of the uses for PCR before we recap everything. So first of all, as during COVID times, and as I mentioned at the start of this lesson, we can detect viral infection at an early stage by taking a small sample of DNA found in, say, the mucous membranes and the mucus produced by them, and we can multiply that so we can get a better idea of what's going on. We can also use it in DNA profiling, which we'll look at quite significantly in our following lesson on gel electrophoresis. It's really important for us for evidence of evolution. This is going to be particularly of note for us as we move into the final topic of the year and when we're looking at fossils and fossil ancestors. We can also use it to assist us with the detection of hereditary diseases, and finally with tissue typing for transplant organs. So what we're going to do now is I've got a little storyboard for us to go through here, and we're going to just piece together this process of PCR, starting from the denaturation stage and working our way through annealing, extension or elongation, and talking about the final product again. So if you've got a copy of this one with you, great. If not, you can just draw this on paper, and we're going to go through step by step what this will look like for the PCR process. So to start with the denaturation stage, as we come down to the bottom here, what we're starting with is our temperature. And during this stage, as you hopefully remember, we heat the DNA to 95 degrees Celsius. Okay? And what this heat does is the heat itself breaks those hydrogen bonds. Just write this down here. Breaks hydrogen bonds between those complementary bases. Now, what happens here as a consequence is that this causes those strands to denature or to separate. And as a consequence, by the end of the denaturation process, we form two strands. We form two single strands, more specifically. So just to recap this, again, the key part here for us right at the beginning is that we heat to 95 degrees Celsius. And what we're doing during this process is we're breaking the hydrogen bonds between complementary bases. What I'll do here is I'll just highlight in here, this is where these hydrogen bonds normally occur, so we're breaking those ones in order to form two single strands. Okay. So we then move on to our next stage, annealing, and during annealing, as hopefully you remember, we're now cooling. Okay, so we were at 95 degrees, but we're cooling down to 50 to 60 degrees Celsius, somewhere in that range. And what happens during this process is that these primers, these DNA primers, anneal or stick themselves onto the complementary ends of those, or the complementary bases, I should say, on the ends of those two single strands. So primers anneal, and what we'll say is that they form hydrogen bonds. To complementary bases. On the ends of the two single strands. Okay. So again, key part here, 50 to 60 degrees. Now for this stage showing these primers annealing, I'm just going to highlight the two primers in blue here so you can see them, and we can see them attaching at the complementary ends of those nucleotides, or at those ends of those two single strands. So we then move on to the third and final stage, extension or elongation, and during this stage we again heat our mixture up, and we're heating it to 72 degrees Celsius. And this is the optimum temperature for Taq polymerase. So I'll just write optimum temp in here. Remember, Taq polymerase won't denature up until 96 degrees, but is working its best at 72 degrees. So what happens here is Taq polymerase attaches itself to the DNA. Polymerase and what it's going to do when it attaches to the DNA is it is going to add those complementary nucleotides and that's going to start from the primer. So let's break this down, let's highlight some of these key components. So first of all, I'm going to try and keep all the temperatures in the same color, so we start by heating to 72 degrees Celsius. Our second key part here is the Taq polymerase attaching. Now, in this diagram, you can see this Taq polymerase wrapping itself around the DNA strand itself. And what this allows it to do is to bring and attach these free nucleotides that we can see. I've just highlighted them in orange here. And they're going to start to attach things from the primers. And we can see the primers on either end here. The Taq polymerase slowly makes its way down the DNA strand, working in one direction and bringing those free nucleotides. And so what's our final product here? Well, again, by the end of this process, we're going to have two identical strands of DNA. And then we're able to simply repeat this process. And we're going to repeat generally 20 to 25 times in order to create a large enough sample. Now when we look at these two original strands, if I draw in some of our primers here, we've got our original strand up the top, and then maybe if I highlight our original strand at the very beginning in yellow here, so we can see that this is our original strand. Oh, I've done it the wrong way around here. There we go. We have our primers binding on opposite ends, and then in orange we'll show this is our new strand that we formed here. So that's a very quick recap of everything to do with PCR and all the steps that you need to know.